Okay, hello everyone. We'd like to get this presentation started. Welcome to our educational webinar titled Using CRISPR-Cas9 to Model Stem Cell Organization and Dynamics, part of our webinar series in the Pluripotent Learning Lounge. My name is Andrew Gaffney and I am a Senior Product Marketing Manager at STEMCELL Technologies. I will be your moderator for this webinar. It is now my pleasure to introduce you to our speaker today. Dr. Ruwanthi Gunawardane is the Director of Stem Cells and Gene Editing at the Allen Institute for Cell Science. Ru leads a group of researchers creating a collection of high-quality gene-edited stem cell lines that are used by the Institute to study cell organization and activities through live cell imaging. Ru joined the Allen Institute after spending 5 years at Amgen, where she developed assays to screen for novel therapeutics in oncology, inflammation, and cardiovascular diseases, and help advance the early drug discovery pipeline. Prior to Amgen, Ru worked at Ambit Biosciences as part of a small team of scientists that identified and characterized AC220, a potent FLT3 kinase inhibitor that is currently in phase 3 trials for AML. Ru obtained her PhD in biology from Johns Hopkins University, where she studied microtubule nucleation in the Drosophila and Xenopus model systems. She conducted her postdoctoral work at the Harvard Medical School, where she studied cell migration and invasion in mammary epithelial cells using 3D tissue culture model systems as a model for breast cancer progression. Let me now hand you over to Ru to begin the presentation.
Hello, everyone, and thank you, Andrew, for that introduction. So I'm very excited to be here today and share with you some of the work we are doing at the Allen Institute for Cell Science using CRISPR-Cas9 and stem cells to study cell organization and dynamics. Okay, so I would like to start by giving you a quick introduction to the Institute, tell you who we are and what we are doing, and then I'll spend most of the time talking about the CRISPR gene editing work we are doing at the Institute and some of the images we've created, like the wallpaper you just saw with my title slide. And then at the end, discuss some of the potential impact and applications that can come from these cell lines and the data we are generating here at the Cell Institute. So for those of you who don't know or haven't heard of us, we are a small nonprofit research institute in Seattle, Washington. We started our research program about 18 months ago, early 2016. And one of our goals is to do the kind of research that typically cannot be done in an academic lab. And the way we do that is using multidisciplinary team science and open science as some of our driving principles. So right now we are about 50 staff members, and they come from very highly diverse backgrounds, including gene editing and stem cell work, which is what I'm going to talk to you mostly about today. But we are we are an imaging institute at the core, so a lot of what we are doing is to understand cells using live imaging. And of course, we also have people working on the computational models to integrate that data we're going to generate, as well as share them with our visualization tools with the community. So why an Allen Institute for Cell Science? So one of the reasons we even exist is this belief that really studying normal cells and fundamental cell biology in normal cells can lead to a better understanding of disease and treatment. And this has been quite difficult to do because there are really no good models of cell behavior, because as anybody working with cells know that they're very complex, very heterogeneous, and quite dynamic. So our mission is to understand first, and then eventually, using computational models, be able to predict cell behavior, starting from normal cells and then working our way towards more pathological contexts. So what do I mean by cell behavior? So just a couple of examples to illustrate this. So I think if you look at the right top panel of images, I think you will all recognize that on the left are interphase cells, on the right is the mitotic cell. And this just highlights this amazing ability cells have to reorganize or repurpose the same organelle, in this case microtubules, to perform a different behavior or an activity, in this case mitosis. But we don't really understand how this happens or when this happens. We understand the proteins, the pathways, but not really how all this is put together. So that's kind of what we mean by a behavior or an activity that we want to follow with live imaging. And another example that's pertinent, since we're talking about stem cells today, is again how going from a stem cell to a differentiated cell type on the right is a cardiomyocyte. Again, these organelles are reorganized or repurposed to perform a very specific function. So how do the cells do that? So we believe that the way to really study this and understand this is to do live imaging and live high-resolution quantitative imaging. So this is what we plan to do to understand the organization of a cell and then the function down the road. And one of our main goals and missions is to share our data and the tools both on the cell side as well as the computational side with the research community. So where do we start? This is kind of a very big goal. So we are first going to start by getting a better grasp of the physical organization of the cell. And what I mean by that is where are the major organelles of the cell and how do they change in relationship to each other and as the cell changes. And this includes, again, through live imaging, studying the position of the organelles as well as the morphologies of these organelles. And what we want to do then is put all this information together and develop a model for the integrated cell. And study this as the stem cell goes from the undifferentiated state through all the way to the differentiated state, as well as things like mitosis and any other process that you can think of that the cell is doing during its usual lifespan. So for this purpose, we really wanted to start with a model system that would allow us to do that, but also would allow us to study as normal a cell as possible. And what I mean by normal is diploid, non-transformed, because a lot of the cell lines that you and I have been working with most of the time are 293s and cancer lines that have aneuploidy and other artifacts that really, I think, have to date made learning fundamental cell biology a lot more difficult, right? So for that, we looked around for a lot of iPSC lines and picked this WTC line, which is actually publicly available, but was generated by Bruce Conklin at the Gladstone. It was derived from a healthy donor episomally. It's been fully sequenced and characterized. Other researchers have used this for gene editing as well as for differentiation into, I think, about 7 different cell types at this point. And obviously, we would like a cell line as genomically stable as possible. But the most difficult thing for us with our goal of sharing everything we do was to find a line that we could openly distribute. And this actually met that criteria. So very happy and excited to use this line. So our goal is, again, live imaging to understand cell organization using stem cells. So we have and continue to create a collection of fluorescently tagged iPSC lines that represent the major structures of the cell. So the way we went about doing that now close to 2 years ago is to engage with the cell biology community and ask them, what are the major structures of the cell that we believe are very important in how a cell is organized and how it reorganizes during the various processes or in development or differentiation or even disease. And this is just, I'm showing you a short list that we started with. The list is a little longer now, and you'll see that in the next few slides. And then we ask them, so what is a good representative of that structure? For example, if we want to look at microtubules, what should we look at? Which protein should we tag? And we got several candidates, and these are the ones that we ended up using. And we also, from the community, asked and looked in the literature to find how to tag this. Should we tag at the N-terminus, C-terminus? What kind of linkers should we use? Always keeping in mind that we do not want to perturb the cells too much. We know that adding an FP is perturbing, but we did not want to reinvent the wheel there. But around the same time, the CRISPR-Cas9 system was getting more and more popular, especially in human cells. So we decided to use that to introduce a fluorescent tag, so for example, GFP, into the endogenous locus of these genes. So we do not want to overexpress the protein, so we are not putting a plasmid there and having the protein overexpressed, because we do believe that will change the fundamental biology of the cell. Number two, we do not want to fix and stain the cells, because that again introduces artifacts that we believe will muddy the waters in our integration and our understanding of how these structures look and work. So that is why we are using CRISPR-Cas9 to knock in the GFP or other FP at the endogenous locus. So what we are creating is an endogenous fusion protein. Once we have done that, we create clonal lines that we perform a great deal of QC on, which I will show you in a few slides, and that we can then use as a reproducible and reliable source for our differentiation assays, as well as for our live imaging. So in the process, our goal was to generate this collection of high quality QC'd FP lines and develop editing strategies, which I'm going to share with you, and the workflows and the QC, not just for our work, for our project of understanding cell organization, but also to release to the community for their own work, since these are structures that are playing a role in almost any study by most people, and having an endogenously tagged line could reveal mechanistic understanding that was not visible or possible before. So we had to overcome a few challenges, because we are working with stem cells. So just to highlight again, careful handling of stem cells before and after gene editing has made a big difference in creating these lines and creating an efficient pipeline of gene edited cells. The second challenge was, unlike just knockouts, inserting a large piece of DNA into the human genome is a very inefficient process. It requires homology-directed repair, which in most cases is 1% or less, compared to the more common non-homologous end joining, which is the preferred pathway for the cell to take. And this is one example. So in this case, we are tagging the gene at the C terminus. So this is showing the last exon. And what we are inserting with HDR is a short linker followed by the FP sequence. So instead of trying to improve HDR, our strategy was to figure out a way to enrich for those cells that are edited. And again, our focus was not to perturb the cells too much. So we really did not want to play around with changing this DNA repair mechanism in the cell. Instead, we just scaled up our transfection so we get enough cells to move forward with our gene editing. CRISPR design comes into play, and I'll show you what we do in the next slide. But I just want to remind you, knowing that tagging this terminus versus that terminus, or which linker, from talking to the experts, even though that might be in an overexpression system, it really helped us move this project along. So doing the research up front really paid off for us. The third part was the design and delivery of these items. So we decided to go with the ribonuclear method protein for introducing CRISPR-Cas9 into the cell. That was again because we felt like having the Cas9 expressed off a plasmid would introduce unknown and unwanted artifacts to the cell that might again make it a little bit less ideal for our work of studying normal cell biology. So we like the idea of just Cas9 protein, because then it would be gone within a few hours, and also potentially reduce both the on and off target effects, unwanted effects. We ended up using multiple CRISPRs just to make sure that at least one of them would result in a gene edit. We did use bioinformatics. So this again, next slide, is a little bit more of a description of that. And if you're interested in our methodology, you can go to our website, allencell.org. We've made our SOPs available through there. So just to give a little bit more detail about the gene editing. So we have found that using 1 kb homology arms give the most consistent knock-in results. We have in some cases tried shorter homology arms, I think about 250 and 500 base pairs. We didn't do that for a lot of genes, but 1 kb always worked best. And since some of these genes are difficult to tag, we ended up just making our protocol 1 kb. And that is what I'm trying to highlight here on the right. So here's the genomic DNA. And if we want to insert the GFP at the N terminus, here are the 1 kb homology arms flanking that, which we incorporate into our donor plasmid. The second point I want to make is that we did deep sequence the whole genome of this cell line, and we're glad we did because the several genes, I think a couple of the genes we've edited so far had SNPs, and if we had gone with the rest-seq available, we would have missed that, and it's possible our CRISPR would not have guided correctly and introduced the edit in that locus. So if at all possible, even if you're just editing one gene, maybe it's worthwhile to do a deeper sequence, or at least at that locus, if not the whole genome. We do introduce sequence mutations into the PAM sequence in the donor plasmid so that the plasmid does not get cut before it can do its job. So those are some lessons we've learned, and we've incorporated it into our editing process. Guide design. There are lots of ways to do this. There's no one way in particular that seems to work better than another way. This is just the way we do it. So we pick guides as close to the integration site as possible. Within 10 base pairs is ideal, although in some cases we have found further away works just as well. We use multiple tools to pick our guides, and those are listed down here. We use three different tools, but you can get 9, 10, 11 different CRISPRs that might work for your locus. But then what we do, I think, which has been very, very useful, is to use bioinformatics to pick 3 guides that then we actually get for our experiments. And for that, we use Cas9 Finder, which basically is not an algorithm that ranks or anything like that, but it looks at sequence homology, and you can define your own criteria for what makes the cut. And if you are interested in what we did, again, you can go to our SOP, and we have a much more detailed description of how we pick those 3 final CRISPRs. At that point, then we are ready to do the experiment. So we introduce these things for each locus into our stem cells, into the WTC line. We use electroporation since we are now introducing RNA, DNA, and protein. And a lot of the other transfection reagents, we've tried them, work for 1 or 2 of these components, but not for all 3. So we've ended up just electroporating, and we are getting about a 0.1 to 4% rate of GFP incorporation into our cells, at which point then we enrich for that to try and get close to 100% of edited cells, or in this particular case, green cells. So this, of course, relies on your gene of interest being expressed, right? Because this requires an enrichment. But you can imagine doing drug selection or any other selection that works for your project to achieve the same result. Our genes are definitely expressed, so we go this route, but we are currently working on methods that would allow for enrichment of non-expressed genes, and we hope to share that with the community in a few months. So our results to date, and I have a table in a couple of slides, but just in general, we've done about 25 different gene edits right now, different loci, and most of the time they have worked. And at first, we were very excited and also surprised that the human genome is this editable, because we are trying to insert tags into very essential genes like tubulin and actin and nuclear proteins. So this is a really good sign, and I think this is very encouraging for people who are thinking of doing this. You should go for it. However, I will tell you that this is very locus and CRISPR dependent. So trying multiple CRISPRs and thinking ahead and designing your experiments will give you the most likely successful outcome. We are most of the time getting monoallelic edits, that is just one GFP on one allele. And this is what we really wanted, because we believe that tagging both alleles might be too perturbing to the cell, especially when we are tagging these essential genes. But I believe using CRISPR-Cas9, the RNP protocol, may have had something to do with us getting just monoallelic edits. The other possible reason is that our cells cannot tolerate two edits at these genes. So we don't know which reason is the why we get monoallelic edits, but this is what we were aiming for. So once we have this enriched population, then we go on to make clonal lines. We are generating about 100 clones per gene at the moment, because we had a lot of criteria we wanted these clones to meet, and I'll go over that in the next slide. So we do a lot of genotyping in parallel to pick clones that are precisely edited, at which point then we do a whole number of QC tests to validate that these lines are behaving appropriately, the tag structure is localizing correctly, and the stem cells still meet all the stem cell criteria. At which point then we use them for our imaging as well as our cardiomyocyte differentiation process. So this takes a long time. So the actual editing itself is probably a week or two. That's not where it takes a lot of time. It takes a lot of time to design this, do the right research, get the high quality reagents together before we even go into the lab. And then of course we spend about 2 months doing various QC. This is really important to us because we are making these cell lines available to the community. So this is a resource we are providing, as well as to make sure the images we generate and the models we create are coming from highly QC'd cell lines. And the way we are doing this, because we don't have a big group to do this, is a lot of parallel processing using some machines, some automation, as I will tell you in a second. So here's an example of how we do our precise editing or our genotyping. So traditionally, when you do a knock-in or a point mutation, you would do a junctional PCR to make sure your GFP or your tag is in the correct location. And we do the same. So we have a schematic here, which might be difficult to see. But since we're using 1 kb homology arms, we have a pretty big sequence too. So we do a tiled junctional PCR assay. But since we're looking at 100 clones, we do use some instrumentation to make this less tedious. And that has really helped us be able to process a lot of cells at the same time. But if you're only doing one gene and looking for a small mutation, this is not necessary. But what I do want to caution you is that if we had just used junctional PCR, there are so many other things that happen at the locus that you are going to miss. And that is why, in our scheme of things, we have incorporated this first assay, which is droplet digital PCR. This is basically another way of doing qPCR, but it allows for absolute counts of your tag, and you basically use like TACMAN probes. So we look for our GFP so we can get copy number out of this assay. We also use a probe for our donor plasmid to see if in some cases the plasmid is getting incorporated at the locus. So that is the major problem we are seeing with our gene editing, with plasmid or parts of the plasmid getting incorporated into the genome at the locus at the same time as GFP. So that is something to look out for, especially if that's getting inserted into a regulatory element that can have downstream effects that you may not realize, but could be detrimental to your assay or to the mechanism you're studying. We also get a very good clue about the clonality using this assay, because if there are cells that are not edited, we can get a ratio of this. So we use ddPCR, junctional PCR, and then Sanger sequencing, which is just to confirm that there are no unknown or unwanted mutations, especially due to NHEJ at the locus. So once we do that, we collect all this information, and then we filter it with our criteria for precise editing mono and biallelic edits. And since we do 3 different CRISPRs, we try to pick clones that are coming from multiple CRISPRs, because that ensures us that we are looking at clones coming from different editing events, which gives us even more confidence of the biology that we are going to image down the road. But let me tell you, that doesn't always happen. Usually one CRISPR wins out. So from 100 clones, using this genotyping criteria, we identify about 5 to 10 clones that we do further QC on. So again, validation of these edited lines is really important to us. So the first question we ask is whether the tagging is precise, and that's what I just presented to you in the previous slide, the location of the tag, the copy number, and the mutation. Second, we ask if the expression of our tagged protein is similar to the untagged protein and whether it's localizing correctly. So we do a lot of Western blots on those 5 to 10 clones and look at the GFP, look for the GFP tagged, as well as looking with a tag-specific, structure-specific antibody to compare the untagged cells to the tagged cells. Once we do that, we also do a lot of imaging, both not just to look at the GFP on the right here, but also with immunofluorescence through fixation and staining to make sure that the GFP tag cells do not have any aberrant localization. And we do this with live imaging too. So then we check whether the genome, the carrier type, is normal, and whether the expression of these fusion proteins is stable over time through multiple passages. And then we continue on once we have fewer clones to look at cell cycle, growth rate, dynamics of that structure. So if you're looking at microtubules, we follow the microtubules over the cell cycle, make sure they're not slowing down or they're showing dynamics that we don't see if we look at the untagged cells. And then we want to make sure that these cells still perform all the functions, at least in terms of the high-level functions of truly pluripotent stem cells. So we look at morphology, make sure the edited cells do not suddenly spontaneously differentiate, which in some clones they do, so we get rid of those. We look for stem cell markers, and I'll show you a slide about that. We do not do teratomas, but what we instead do is directed differentiation of the 3 lineages using the stem cell technologies kit. This is early differentiation and look for markers, and we've done the EBs as well. And then since we want to study cardiomyocytes, we do terminally differentiate this into cardiomyocytes, and that really gives us even further confidence that these cell lines, even though they're edited, can perform these functions that we expect stem cells to do. So I'll show you what that workflow looks like. I'm not going to go into great detail. There are about 17 different assays we do. So like I mentioned before, we do these genotyping assays right up front in a 96-well format because they're relatively easy to do. And then we do Sanger sequencing on a subset of them, and from then on, we start doing the more laborious but really essential assays on a select number of clones. And our goal is to pick 1 or 2 final clones for scale-up, for our microscopy work, as well as for distribution. And so, like I mentioned, we do imaging, both live and fixed. We do Western blots. We do sequence for the final clones the full allele to make sure that is as expected from the Sanger sequencing we did right up front with genotyping. We do karyotyping 2 or 3 times during this process because we do know that things can happen as you expand these cells. And I mentioned these 2, but we also look at off-target. The sites that we find with Cas9 Finder, we then go back with these 5 clonal lines and sequence and make sure. And just to summarize that, we have yet to find 1 clone out of the 50 or so clones we've done that has the off-target mutation, and we've looked at about 10 different sites for each clone, around 500 sites sequenced today. So what we've learned is that if we pick the clones that pass our precise editing, which only 25% of our clones do, about 50% of our clones end up having the GFP, but about half of those also have plasmid incorporated into them. So we get rid of all of those. So usually it's like 1 in 4 clones meet our criteria. This is very gene dependent. In some genes, it's 90%. In others, it's 10%. So this is an absolute average over 20 loci, so that you know. But our clonal recovery is pretty good. Again, we take a lot of care in how we grow this and culture this. And great. So my point is, if we do this upfront work of genotyping, it ends up 4 out of these 5 clones usually pass the rest of the QC. So that is really good news. So there are some trends and observations and lessons we have learned through editing over 20 loci. Again, I'm not going to go into this at great depth, but I've mentioned these things already. But I highly encourage you, if you want to know more, we've done a retrospective analysis of all of our gene editing over the last year, and it just came out in MBOC this month, and that's the citation down there. So on the right, I'm just showing you how this is very gene dependent and very CRISPR dependent. This is the editing efficiency as measured by FACS. But the good news is, even if it's 0.05%, we can get an edited line out of that. And stringent QC is essential, especially our 3-step genetic screening strategy has really paid off. So we took some of our clones that failed our criteria, but would have passed the junctional assay, which are shown on the right here in yellow. And you can see here, if we had just done junctional assays, a lot of them would have actually not been precisely edited. So that's just a warning, and it depends on what you use these cells for and what kind of assay. But just to know that if you're creating a double-strand break, there's a lot that happens at that locus, so to keep that in mind. So again, just an example, we confirm stem cell markers using flow. We use cell surface markers. These are the ones that should be expressed in pluripotent stem cells, and a marker that should not be expressed. And we have certain criteria set. And this is the trilineage assay, where we use a different media for each lineage, and we take the cells out at these different time points and look for markers that are representative of that lineage. And that's looking really good so far, and we have incorporated that into our QC at the moment. So as I mentioned to you right at the beginning, what we really want to do is not just study how these organelles behave in the stem cells, but how they change as the cells become cardiomyocytes. So we've started now developing protocols in-house to differentiate these tagged iPSC lines into cardiomyocytes, and that's looking really good. These are just to show you 2 examples of our beating cardiomyocytes. On the left, the cells are beating, but they're more twitching, and the other cells are beating really hard. So there's a very diverse phenotype, so that is expected, and the protocol we are using is on the right. So we've done this now many times for our edited structures, and it's pretty consistent. And again, that's in our paper, and we are getting to see the beautiful morphology and the organization of some of these cardio-specific structures in these cells as well. So this is the list. This is more of the current list. So we have about 25 structures we have tagged to date. On the left is the structure, in the middle is the gene we are tagging, and then the FP and our status. This is very much a pipeline. So we've tried to tag about 28 structures. 25 have been successful. 17 out of the 19 that we've looked at so far have passed. This guy failed because of a karyotypic abnormalities. So we dropped that one. Luckily, we had another gene for that structure. Vimentins, we failed because our tagged cells look a little different from the untagged cells. We don't understand why. It could be quite normal. We just don't understand intermediate filament biology in stem cells. It's really hard to know what's right and what's not. So we paused that, and we do not release any cell line that we don't believe the cell biology is looking as expected. And that is kind of difficult because a lot of this is not known in stem cells. So this is just to show you our unedited cells, where the genes we are tagging lie in terms of expression profile. And the scale is just 2 samples. To the right or above is just log folds of RNA transcript level. And as you can tell, the top right ones are actin and tubulin. Those should be relatively easy to tag. And genes down at the bottom here that we can't see very much expression would be difficult to tag only because we're using FACS enrichment, which requires expression. But surprisingly, some of those genes that have very low expression and very focused localization, we were also able to tag. The ones in black are the ones that we were successful with. The ones in red, we were unable to enrich for. So we don't know whether that was expression level or the editing. These two here are cardio-specific structures. Again, like I mentioned, we are very excited that we think we now have a protocol to do non-expressed gene edits. And those have worked, but we need to validate that before sharing that too much. So what's next is working on more genes that are not expressed in stem cells, such as cardio-specific markers. We are going to take a little bit of deeper dive into nuclear proteins and some signaling proteins, because we believe those are going to be important to understand the organization and the dynamics of the cell, in addition to these major structures on the left here. For our modeling group, they really want 2 structures tagged, one with red, one with green, so that they can understand the correlations, and we are working on that as well. So like I mentioned to you before, we want to make these available to the community, and we are doing that with Coriell. So we have made 11 of these cell lines available through Coriell. And those are listed in green here, and this is when we release them. So there's another set that we're just getting ready to release in a couple of weeks. Those are, I think, personally very interesting, like the centrosome. If you're studying any of these structures, please do go and look at that. Come to our website to see the movies, and this is what's coming up next. But maybe even more exciting is that we are making these plasmids, which we spent a lot of time thinking about and designing and getting the information from experts through AdGene. So if you want to make these lines in your own favorite line and not use iPSCs or use a different iPSC line, you can do that. And the methods, again, the paper just came out, and it's also all on our website. Just to show you, if you go to allencell.org and go to the cells, we have a cell line catalog, and you click on one of these links, and we've put a lot of information here. You can look at the Z-stacks, the time lapse. For any cell line, there are links to Coriell. And then we also are sharing with you our editing design for each locus. So, and even the CRISPR that ended up giving us our final clonal line, so that we really want to empower the community to do this. And that is part of our goal. And we summarize all that QC. We are doing the 17 different assays into what is like a spec sheet, and we provide that to Coriell to go with any of the vials that's being distributed. So we scale it up, we do all the work, Coriell is distributing, just to make that clear. So I'm going to spend the next 10 minutes to show you what these cells look like. This is what gets us cell biologists very, very excited. So the stem cells, those of you who are working on this, they do not grow as single cells. They grow as a colony. They actually have this amazingly beautiful epithelial morphology and characteristics. And on the right here is just fixed and stained cells, where the red, the cyan is the plasma membrane. The blue is the DNA, and you can see how big these nuclei are. And these cells are quite big. They're 10 to 20 microns in diameter and about 10 to 15 microns high. And on the right, I'm going to show you a movie, a Z-stack. So these cells are grown on Matrigel, on tissue culture plastic, but for our imaging, then we transfer these cells to glass, but they still have the Matrigel coating on there. So what you're looking at is a confocal Z-stack series taken on a Zeiss spinning disc microscope. And these are our actin tagged cells. And you'll see the stress fibers at the bottom, and as I move to the top, you'll see the beautiful cortical actin belts. And this will loop through twice. Stress fibers, you can see the black holes, the nuclei, and the cortical actin. So this was one of the first ones we imaged, and we were still amazed. This is not overexpression. This is endogenous actin expressed from the cells. And you really don't need super resolution or any kind of really high end. I mean, this is somewhat high end. And there's no background because there's no staining artifacts. So you can get a lot of information from just really good confocal microscopy. So right at the beginning last year, we were doing some transient transfections because we were just getting started. And this is one of our mitochondrial proteins, TOM20. And as expected, there's a lot of overexpression artifacts. Really, you can't see very much here. And there's a lot of cells in the background here that never got transfected. On the right is our gene-edited cells. Again, a Z stack going from that's the top, bottom to the top. And again, we were just amazed by the detail here. And we know mitochondria form dynamic tubular networks. We just did not see that on the left. But on the right, we are seeing what we have been taught, how mitochondria look and behave. And so we're very, very excited. So we really believe working with normal cells and endogenously tagged cells is a game changer to understand these kind of fundamental cell biological questions. So here's our type junction. This is Z01 tagged cells, again a Z stack. In the bottom, there's no Z01. On the top, you can see it in the intracellular junctions. And on the right is a lower magnification, but it's a time lapse. And I think we imaged this just a few weeks ago, and I had no idea. So you can see how dynamic this colony is, and all colonies are. And you can see maybe on the top right, my pointer may not work. Some of these tight junctions release. It turns out the dead cells in the colony are being ejected. And that's what you're seeing with those, some of the stain going away and coming back. So I could spend an hour showing you all our live images from these 20 structures, but I don't have an hour. I'll show you a couple more. This is my favorite, having worked on microtubules before. This is the colony undergoing division, and these are tubulin tagged cells. And you can see how dynamic and how active these colonies are. There's a lot of cell division going on. You can see how different the cells look at the edge versus the middle. And this is a higher magnification time-lapse movie. And you can almost see the individual microtubules, and you can see that cell division. Again, not super resolution microscopy here. It is just very clean because there's no background, and it's endogenous expression. Here's lamin B1. This is the nuclear envelope. On the left is the colony, and you can see a cell that had just undergone division there. And on the right, I'll show you a time lapse. And if you watch those two cells dividing on the middle and upper left, we were actually quite surprised by this because I always believe the nuclear envelope completely breaks down, and it looks like it does not. And now that we've talked to more experts, it turns out that's actually true. The nuclear envelope may not completely break down, and this is more reminiscent of lamin B1. And those are the kind of observations that we think are going to be really impactful for studying all these dynamics of the different structures. So on the left is ER. And now we are noticing similar observations, and the ER and the nucleus are connected, so it's not surprising. So we are very excited, and what we are doing now is tagging both of these in the same cell, one with red and one with green, and we are going to watch live what happens. So those are just images we created as part of our QC and got us all excited, but there's a whole group at the Institute working on automating this so that we can create large-scale data, and this is a big deal because it's not just about creating the images, but also processing and extracting these images. And we're currently working on creating this microscopy pipeline And then what we want to do is extract this information. So since each cell has only one structure tagged, our goal is to integrate all those structures into one cell and create an integrated cell model. So we are extracting that information, and also all these cells we are staining with a nuclear and a plasma membrane dye, so that we get positional referencing, so we know where the mitochondria are related to the nucleus and the plasma membrane at all times, which really helps us integrate all this information to create this, this integrated cell. So this is what our modeling team is working on. They're using computational tools and deep learning tools to integrate, encode, and decode this information. And they're also trying to figure out how to incorporate all the variability. So no two cells are alike. I think we kind of knew this before, but it's amazing now that we've imaged thousands of cells. No two cells are alike, even though they're clonal. But I think we believe that these cells are telling us something, we just don't know what it means. So by doing this at a high enough scale and using these computational models to help us do things the human brain cannot do, and also studying individual cells, cells going through cell cycle, differentiation in 3D, introducing known agonists and antagonists, I think we can kind of profile what a typical cell undergoing a typical process should look like. And we're hoping to have something called a cell clinic, where you bring in your cell with your cell morphology, and we tell you, or you can go and find out what the most likely state or stage that cell might be in, based on the information we are generating and through these probabilistic models we are building. So with all of this, our goal is to figure out, back to our lofty goals, is there a master organizing the cell? What are the hierarchies of organization? And what drives reorganization as the cell goes through these various processes, including differentiation and cell cycle, and absolutely establish correlations, and hopefully this be a hypothesis generator, not just for us, really for the community. And this is where our Allen Cell Explorer comes in. This is how we want to help people look at our data, go in, and we have a visualized, and I'm going to show you an example in the next slide, a multi-scale view of the cell, kind of like the Google Earth, but the Google cell. And we will also share our computational models, and we're trying to do as much open source software as possible, so anybody can come in and look at the data and do your own search and go from there. And really one of our main goals is to go from the cartoons that we have all learned from and go to much more data-driven representations of what a cell really looks like. So this gets back to the wallpaper I started my talk on. So these are some of the images we've generated. So you can go to allencell.org. I think there are like 6,000 images there. And in the red, again, is the plasma membrane, blue is the DNA, and the white are the different structures we've tagged. So if you look to the middle right, you can see that cell undergoing cell division, and you can see the spindle, the microtubules. That is definitely a tubulin tag line. This one is a Z01. You can see the apical staining. It's not staining, sorry, localization. I don't even remember what these ones are, but you can go and pick your structure and look at the cells if you are interested. And I kind of want to end by saying that using stem cells, there's this amazing power to study this continuum of cell states that are important in development, in disease progression, and we are just studying these iPSCs going into the cardiomyocytes, but there is this naive state of iPSCs that people are starting to look at that are very different epigenetically and morphologically to the primed iPSCs that we all use in human cells. And we are just studying right now the iPSCs and the cardiomyocytes, but we hope to do is to do live imaging as the cell transitions, as I mentioned several times. But what I didn't mention to you, because I didn't have enough time, is that we are doing RNA-seq, single-cell RNA-seq at all these time points too, to help in building our cell models from the genotype to the protein and the localization and the dynamics. So we're kind of thinking of it as a way of atlasing the stem cell to really still help identify some master regulators of these changes and eventually be able to predict the state of a given cell. But there's an amazing opportunity for us and for the community here, because we have created these tagged lines to differentiate into other cell types, to do genomic screens, to do a CRISPR-I screen or a chemical inhibitor screen, to see if you inhibit one pathway, if you're studying just mitochondria, but you can look at the ER, you can look at the Golgi, you can look at the nucleus through live imaging, and maybe get a better, higher level understanding of what the implications of this are, especially if you introduce disease mutations, take it into 3D. I think the sky's the limit here, and so we are very, very excited about the opportunity we have internally and to work with the community to advance the field there. And I think this is going to create better models for development, understanding development and disease. So we have multiple collaborators right now working on 3D organoids, for example, the kidney, and trying to learn glomerular function in polycystic kidney disease. If you go to our website, there's a short movie on that. So we're very excited that people are starting to use the lines and to really understand mechanistic biology as well as better disease modeling. So hopefully I've told you about how we are doing our CRISPR editing, shown you some images to kind of show that the power of live imaging and how we are sharing the tools and the data and the potential impact of this work. I have to thank this is the Cell Institute team on the right here. And I have to thank my team, the gene editing team, who have done an amazing amount of work in just over a year. And these are the folks who did that live imaging that I showed you about, led by Susan Rafalski. So thank you for listening. And I have to give big thanks to our founder, Paul Allen, for his vision and his support that has made all this possible. And with that, I'll thank you and stop there and take any questions.
Okay, thank you very much, Ru. That concludes the presentation segment of the webinar. We'll now begin our Q&A session by addressing the written questions that were submitted throughout the presentation. So let's start with our first question. Could you improve the problem of backbone integration by transfecting the piece of donor DNA without the plasmid backbone, and that's either synthetic DNA or restriction digest, for example?
Yes, so that is difficult to do, and that might get incorporated too. But that is a very good question. What we're trying to do is single-strand DNA. I don't know if most people have heard, but people are now able to make large pieces of single-strand DNA. So we are looking into that because that will, number one, significantly reduce the incorporation rate. It may also reduce the need for 1 kb homology arms. So if we can shorten that, then we can also screen much easier because we don't have to span now about 3 kb. But that is something we're actively trying to improve. We just didn't even realize it till we started screening these clones.
Okay, thank you. Second question, how do you transfect the cells? Are there any methods that you've tried that were better than others that you've tested? And also, what's your transfection efficiency?
So we use electroporation. So we did try, multiple different methods. I can't remember off the top of my head. Lipofectamine methods. We tried about 9 different transfection methods and found that in all those cases, either the DNA goes in well or the protein goes in well. But remember, we are putting in RNA, DNA, and protein. With the electroporation method, we can get up to 50% transfection. But there is some cell death associated, so that's not ideal. So we just do a lot of cells to end up getting enough cells to then do our enrichment with. So about 50% transfection.
Okay, thank you. Next, are the clones derived from single cell deposition? And if not, how do you ensure clonality versus getting mosaic colonies?
Yeah, good question. So we do not do single cell isolation of facts. They don't survive, although there are some media that we can try and improve that. What we and others have done with this cell line is to get the enriched population and then seed them at clonal density. So that turns out to be, again, you'll see if you look at the protocol, about 10,000 cells in a 10-centimeter dish. And then we go in and manually pick individual clones, right, that are separated from other clones. So we can never 100% be sure that they were derived from a single cell. I actually believe that's not even possible with the cell division rate. What we can say that they're genotypically the same with ddPCR, because you can do certain different probes and look at ratio to a reference genome. So that's how we look at clonality. And then, of course, when we do our imaging, we look at brightness. And as a mixed population, we see different brightness. And then when we make our clonal lines, we pick clones that look as homogeneous as possible, and that genotypically and by Sanger sequencing, from what we can see, it's pure.
Okay, thank you. The next question we had asked a few times, so I'm going to combine it into one. Have you found that the tag that you use affects cell behavior? For example, cell growth, differentiation potential, and that's linked in with, is there GFP toxicity?
Yes, no, we have not, interestingly. So we use a GFP that has been used in the literature and by people for a very, very long time. A lot of people made suggestions on FPs that are newer. We went with the old-fashioned GFP that has been used for a very long time. So it's fully vetted. No, so for every cell line we've generated, we are looking at multiple clones. And like I mentioned several times, we are looking at the unedited cells, right? So we're looking at mitotic index. We're looking at length of time at different parts of mitosis. We do growth rate, so we do a 48 and a 72-hour cell growth rate with the ATP readout. And in this clonal line generation process, we do a lot of culturing, so we always keep an eye on whether some clones grow faster or slower. That does indeed happen when we are at like the 24 clonal stage. And then the clones that slow down or speed up also have other issues with them. They start to spontaneously differentiate sometimes. So those end up getting dropped, but we don't know for what reason, because they just don't meet several of our criteria. But in general, the two final clones we pick, we do not believe, or we do not see any observable effect. That does not mean if we did a functional assay, for example, with the microtubule line, if we do like certain dynamic assay, that we wouldn't see a difference. We really do think a GFP putting a tag in there has to be somewhat perturbing. We just haven't seen it.
Okay, so we actually had a question from a researcher who's currently using two of your lines already, who asks, I'm doing the actin polymerization assay. Can I use the GFP antibody to detect G and F actin in your cell lines?
Yes, you should be able to. Sometimes we use that, not for actin, because actin is one of the most highly expressed proteins. We haven't had to. For some of the lower expressed proteins, we do add GFP just to check and confirm with the unedited cells. So, but I don't believe we have done that yet, but I don't see why you couldn't. But you should email us about that one, because I can check with our Western blot assay development group.
Okay, thank you. And a comment from somebody who is interested in the imaging side of the Allen Institute. So you said that you mostly use confocal microscopy. Do you have any plans to move into super resolution, for example, palm, storm, or lattice light sheet?
Yes, those are all things being considered. So we do have an Aries scan, which is about 1.2-fold better resolution, I believe, than what we're getting with the spinning disc. But those things are being considered. It depends on the question we want to ask. So we are always keeping those in mind and are in conversations with vendors who are building more pipeline compatible instruments for those, because a lot of those things that you just mentioned are mainly used by institutes or labs. You're working on one or two things. We have a pipeline going where we're creating lots and lots of images and using stem cells, which are a little bit more finicky than some of the other cell lines. So we're trying to figure out what the best modality is and having multiple options depending on what we want to study, because certain dynamics are going to be faster than others. So some of it comes down to timing. And these cells are quite large, so it takes a while to get a Z-stack because they're about 15 to 20 microns tall. So these are all the challenges we are now having to deal with working with stem cells and looking at dynamics, and then start to work on cardiomyocytes, which are beating, but we still want to do live imaging. So, but nothing's off the table. This is just what we started with.
Okay, thank you. And along the same lines, what microscopy system do you use for your live imaging? Is it automated?
Yes, so it's not automated in a truly, automated fashion. Some aspects of it are automated, and that's what the microscopy group is working on right now. Most of it is currently manual, and we're using the Xi system.
Okay. Would you also recommend CRISPR-Cas9 to clone a his tag into a gene in order to do a pull down, then mass spec?
Absolutely. Any tag. And his is much smaller than GFP, so it should work. The more important thing is to make sure you tag it at the right place, right? So it does not affect binding to its binding partner. So if it's important in regulation, but if you just, especially if you want to do a pull down to see what complex it's in, but it should be very feasible.
Okay. We saw that you use automated handling systems for cell culture. What kind of systems do you use, and in what stages of the workflow do you use that automation?
Good question. So my group does not use automation, the stem cell and gene editing group, but the microscopy group, just like I was answering the previous question, starting to build automation into their workflow. And what they have is a Hamilton. I don't know the model, but it is a Hamilton robot that has an incubation system. And what they're using it for right now is to seed the cells onto their glass imaging plates. So that they're very consistent. What the robot offers is the kind of reproducibility and consistency that a human hand cannot. So that's what they're doing. The actual culturing of the cells still done manually, but we are, of course, starting to think about that to make, at least for the microscopy piece of it, to make this as automatable as possible.
Okay. Next, what does the Allen Institute currently do or plan on doing with naive pluripotent stem cells, and how might that be different to primed?
Yeah, good question. So the naive, we have not done anything on this. It is just one of our interests because we do want to study the cell in various states, and we were always thinking towards differentiation. But seeing the literature and the naive iPSCs, our thought is why not even go backwards? It just allows us to broaden the continuum of cell states. So we're still thinking of it from an organization point of view. Where are the organelles, especially if the naive cells are a little bit more in a pluripotent state, And we know, for example, the mitochondria in particular look very different and perform very differently in naive cells. It's one of the structures that's actually being studied. But what about the other structures? So if we have a tagged line, and we have 20 now, close to 20, you can de-differentiate them into naive cells in addition to differentiating them into cardiomyocytes. And then we can have this broad spectrum of images to use for our modeling. So it's just very much a vision right now. We haven't actually done any work, but we have people across the street at University of Washington studying naive cells. So this might end up just being a collaboration.
Okay. You mentioned using ddPCR, fragment PCR, and Sanger to characterize FP integration sites and frequencies. Why don't you use next-gen sequencing as a single test to deliver this information?
That was more about the resources within the Institute. Remember, we have only been doing this for about a year, so it was just a bigger purchase and bigger resource that we wanted to sign up to do. The outsourcing of it would have taken too long. So it just made sense for us to do this with 3 or 4 people. It was just much easier. But it is something we are constantly thinking about, whether to bring in that ability in-house to make this fast. And right now, the way we see it, it's not going to be fastest, and that's why.
Okay. How big is the plasmid that you use, or what is the limit or the size of the plasmid?
I don't think there's a limit to the plasmid size because we're just electroporating it. I think it's about 5. It's puck. It's about 5.7 kb. We have used it to insert up to 3 kb for another pilot project. So I can't say for sure, but I'm assuming that then it was around 8 kb.
Okay. And how do you identify off-target effects in the genome?
Yes, so multiple ways we are doing that. One way is the Sanger sequencing. So we look through Cas9 Finder and look for sites that have certain matches to this, the region that we are editing in the seed region and the non-seed region. And then we go and Sanger sequence those. But if you're aware of the literature, it's not really clear people understand where that cutoff is for the sequence match for an off-target cut. So we are now doing RNA-seq and exome sequencing of our final clones, the 2 clones. And we are then going back and looking at a longer list of potential off-target sites. And so far, we've still only looked at up to 3 to 4 mismatches. There's still none in our first 10 lines. And then we are looking just globally at the RNA-seq levels to see if there are any changes in transcript levels, especially in the gene we are targeting and also in the predicted off-targets. And this work is just still ongoing because it took us some time to do the RNA-seq and exome-seq, and we're getting that data right now. We hope to share that as well through our website when we do have it. But just from the Sanger sequencing and just looking at our first 10 lines through exome sequencing, we are still not seeing anything.
Okay, thank you. Do you control oxygen to physiologically relevant levels around your iPSCs and your differentiated cells?
Not yet. So that is, yes, some people do use hypoxic conditions for growing the stem cells and especially for neuronal differentiation. But for cardiomyocyte differentiation, most people are not using that. So we have also stayed away from that. But it is an option down the road if we find that that increases certain consistency, reproducibility. We would love to know that. Nobody has actually told that to us. So we have the option of doing it, but we are currently not doing that.
Okay, just two more questions before we finish. Have you integrated the edited cardiomyocytes in vivo or even the edited iPSCs?
In vivo, no, we have not.
Okay, and finally, when you move to the disease cell types and compare them to the healthy lines, what is your next plan after that?
So we have some parallel plans. We haven't decided what's really next yet, because we are still very much in the middle of generating the data on the stem cell lines, and then we have a lot to do this for the cardiomyocytes. Part of what we want to do is introduce disease mutations. So it would make sense for us to start with cardiomyopathies, since we are making cardiomyocytes. So that's something that's very much within the works. We are very interested in going 3D because that is much more relevant in a way. But we just don't have the capacity right now to go there. So the way we are doing that is through collaborations or encouraging the community, which is one of the reasons we are sharing these lines, to use it in their models in parallel or as a complementary approach to learn more. And then if we find that there's something novel there or that is related to what we want to first understand, which is how the cell physically changes, what are the dynamics, then we can incorporate that into our plan. So right now our plan is to do these things through collaborations, making these cell lines available, and then focusing first for us to finish up our work on organization across multiple states. So hopefully that answered your question. But introducing some disease mutations either by us or with a collaborator is kind of our next step. Oh, and one other thing, and we are also doing some adding certain inhibitors into our cells that are obvious ones, not a drug screen, but more of a characterization screen to learn more about the dynamics of the various organelles with known inhibitors and agonists.
I really want to thank everyone for attending, and I'd especially like to thank you, Ru, for your time and this excellent presentation.
Thank you very much. And please, if you have more questions or are curious about the Institute, you can contact us. I think the information is on allencell.org, and I highly recommend to go take a look at our cells and our movies.
Fantastic, thank you. So thank you again, and goodbye.